My name is Manju Singh. I am working as an assistant professor in HR group of institutions, Ghaziabad, in pharmacy department. Today I am going to teach you industrial pharmacy second with code 702T. But today's topic is unit 3rd, topic name Biostatics in Pharmaceutical Development. So, let's start. So, first of all, everyone should know what is biostatics. Again, biostatics is made up of two words, bio plus statics. As we already know what is statics means. So, biostatics is made up of these two words, bio plus statistics. Statistics is related to maths or calculations, and bio is related to biology. So, the calculation of biological terms or biological studies is known as biostatistics. We can also see the definition of biostatistics. Here it states that statistics is the science which deals with the collection, tabulation and classification of numerical facts as the basis for explanation, description and comparison of the phenomena. In also the collection, tabulation, classification, organization of numerical facts as the basis for explanation, description and comparison of various factors. Here, biostatics is the branch of statistics concerned with mathematical facts and data related to biological events. It means various biological events which occurs around us or inside human bodies. If we want to make a mathematical calculation of the facts, so we use biostatistics. Biostatics is also known as converting information into figures. It simply means if there is any information for the biological events, if there is any information related to biological events, so the statics, by using statics in which we do mathematical calculations or tabulation or collection or organization, it converted the information into figures or tables. This is known as biostatistics. For a very common example, if there are 100 students in class 5th and among 100 students only 50 students pass with first divisions and rest students passed with second division plus third division, and Among the top 50 of first division, only 5 students passed with distinction. These are some information, and we have to represent this information in table form or mathematical figures. So we use statistics. Likewise, if there is an example, like if there is a health checkup or blood donation camp inside a school or a campus. So in a health checkup, there was a result that among the ladies, 85% ladies are with are suffering from anemia or less hemoglobin, the report says. So this report is related to the biological event, because we're talking about the hemoglobin of ladies. So this is related to biology, but we have to represent this fact in table form or in figure. So when we convert this information in figures, this is known as simply biostatistics. There are various formulas and various tables for statistics. This was a simple information, how many ladies were suffering from, but there are many complex informations which we have to convert into figure. So for that statistics is used, because this was a simple information. We can write down 1, 2, 3 or that many ladies were suffering from hemoglobin, but when in next slide we will discuss about this. So next, first of all, we should talk about the history of biostatics, how biostatics came into existence. So here Sir Francis Galton is known as the father of biostatics. Sir Francis Galton is known as the father of biostatics. How? Because he was the first scientist or first person who introduced statistics into biology. He concludes the inheritance of intelligence and various other factors and make the information or facts into figures by using statistics. That's why he is known as the father of biostatistics. Next is, why do we have a need of biostatics? Biostatics is, or simply we can say, statistics is known as the method of comparison between two or more things. In schools we have studied mean, mode, average. We make a table and put figures according to these. So these are the comparisons. So in statistics we do comparison, and in biostatistics we do comparison of two or more biological events. So for example, there is a survey in a local area, how many people were suffering from coronavirus. So the agency who on the task of surveying there has concluded that 40% of people above age 50, below age 18 are suffering from coronavirus. Next, this is a one fact. Next is, in same local area they conclude that people who were aged above 60, and among them at least approximate 80% people are suffering from coronavirus. This is a next fact. Third is ladies with age range from 15 to 35 are suffering from coronavirus, and their percentage is 35 percentage. Next, again, these are three different factors in the same local area. Again, there is a fact that persons suffering from diabetes are more prone to coronavirus, and among them 87% who were suffering from diabetes were also suffering from coronavirus. So, this is the information gathered by the survey team in a local area. Now, they have the collection of the information, but the next point is how will they represent this data to the authority. So, they cannot represent the data in simple forms like 1, 2, 3 or that is. So, they have to make a table form of this data so anyone can easily understand. So, they will form a data like this and make a chart: diabetes patients, girls, women's, man's, age factor, ages, below age, above age. So, there will be a different chart. So, this chart will be made with the help of statistics, because there are many factors which included in this, like ladies suffering from coronavirus are different, but who were suffering from diabetes, this will also include ladies and children. So, the diabetes factor is different from the elderly persons, from the young children and from the ladies. And ladies are also included in diabetic patients suffering from coronavirus. So, this is a complex chart. So, this chart will be made by using statistics. So, here we will take help of biostatics. Number second, this is type of biostatics. First of all, next point is genetics. According to Sir Francis Galton, who was known as the father of biostatics, first of all, biostatics introduced in the genetics. In genetics, as we have read in our 12th classes, there was a genetic study on P. sativum and various others. In genetics, we do biostatics, and like if there is a female with brown eyes and male with black eyes, so what will be the fourth or third generation? In these, we use statistics formulas or inheritance of genetics, inheritance of various other factors, intelligence, your colors, skin colors and other random facts. So, in this we use biostatics. Next, in pharmaceutical industry, we use biostatics very much. Like first and prior to this, first of all, we do statics in our research projects in colleges. Like if you are doing M.Pharm, M.Doc or any other project work, when we do our project work, we conclude some facts and figures like results, but the results in figures. So, we make tables We make charts. If for example there is a dosage form and we have to check the bioavailability, we make plasma concentration versus time. We make charts. We make different dosage forms like 1:2, 2:3, 1:4. So we check the bioavailability of these ratios by making biostatics with the help of statics. Next, in pharmaceutical industries. In pharmaceutical industries, there are clinical or preclinical or non-clinical trials conducted. So, the non-clinical trials on animals and their results are collected and organized in tabular forms with the help of biostatics, and also like this the clinical trials on humans and the results of clinical trials are arranged according to biostatics, because the result is comparatively very different and it is a complex figures. So, we have to use the statics to represent the data. For example, there is a drug, an anti-diabetic drug. There is a diabetic drug, and we were doing the preclinical studies on rodents. So, we have to take the data or collect the information of the diabetic drug on rodents and report the result in table forms, in graph forms, not in information in the description. We have to represent the result in statics forms so that the authority to which we are representing the data can easily understand. As for example, there are we were doing a survey for a survey of people having cell phones in a rural area. So, most of the people are uneducated there. So, what will the survey team do? They will make a poster and make a graph that 80% people are using cell phones. Because people are uneducated, they can easily understand by graph that people are using cell phones. So, in spite of giving a brief description of this information, we simply use graph or tables with the help of statics. Next, in hospital records. In hospital records, everyone has shown there is a record of every patient. Maybe sometime it is in descriptive, but mostly the record is in table form, that date, dose, patient response. The most popular patient chart, most popular chart in hospitals, and after seeing the patient chart, everyone can know, or the non medical people can understand what is the stage of the patient. He can see the date on which he admitted and the doses he is taking and his result and his condition, all are represented in tabular form, so they can easily understand. There is also a record book of every patients and record books of every treatment and their doses and their pharmacological actions. So, by studying these, doctors, physicians can easily understand the condition of patients. So, biostatics is very helpful in hospitals. also helpful in other areas, but biostatics, by using biostatics, the information can be easily understandable. Next, in types of biostatics, and before types of biostatics, I want to include some more topics like in nutrition. In nutrition, suppose the team is conducting a survey on nutrition of ladies in a hospital. So, they conclude that mostly ladies, or 75% or above 75% ladies are anemic. So, they make like in pandemic era, in this pandemic era, many surveys has done for the key if the covaxin is effective or Covishield is effective or other vaccine is effective, the survey has done, and the information related to surveys is represented in statistical form. Next is type of biostatics. Number one is inferential biostatics. In inferential biostatics, here it is the method of generalizing a larger group. Inferential means, as the word implies, inferential, it means it inferences a lot. It means the method is used when we are generalizing a larger group. The group is large, so many people. The generalization is based on the information about a sample. The sample is considered to be similar to the population. Descriptive biostatics. It describes what data is or what the data shows with the help of graphical or tabular representation. Here is the main difference between these two types is inferential biostatics is used for a large group and based on information about a sample, and the sample is similar to the population. Next is descriptive biostatics. This is for large group and this is for small group. Next is stages of biostatics. What are the stages of biostatics? So, there are various stages. We can say this stage. The step 1. What is the step 1 of biostatics? Step 1 is, first of all, we do drug discovery. It means we have information of thousands of compounds and we discover our drug from these 1000 compounds and make a table. Like there are many drugs and we exclude our drug from here by using biostatics, and the time period taking in the drug discovery is 4.5 years. Step 2 is preclinical testing. Preclinical testing means animal testing, and time period is 1 year. In these preclinical testing, we conclude the results in 1 year, and after that, after approval of this, we do clinical trials from 1 to 10 compounds, and time period is 6.5 years, because there are various phases, 4 phases, phase 1, phase 2, phase 3 and phase 4 of clinical trials. Step 4, after clinical trials, we send the data to the FDA for approval, and it takes approx 1.5 years. Step 5, after approval of drug, we launch the product into the market, and time period is 6 months. It means we also do post market surveillance. Next is clinical research protocol. How do we do clinical research? It is a complete description of and scientific rationale for research activity involving human subjects, and protocol means rules and regulations, laws to clarify the research question, compile existing knowledge, formulate hypotheses and objectives, to decide about a study design, clarify ethical considerations, apply for funding, and have a guideline tool for the research team. Parts of protocol. What are the parts of protocol? So, we have introduction, abstract, objectives, background, rational, eligibility criteria. Study design are like there are various experimental designs in industry like random trials, blinded trials and many more. Criteria for evaluation: study, treatments, clinical assessments. Next is clinical laboratory measurements. Evaluation by visits, adverse experience reporting and documentation, discontinuation and replacements of subjects, protocol violations, statistical section, administration, ethical, regulatory considerations, reference appendices and publication. Next is concurrent medication. All subjects should be maintained on the same medication throughout the entire study period. Standard therapy for specific disease is allowed except for treatment noted in the exclusion criteria. And now dosage regimen. In dosage regimen, we do what is dose, route of administration, schedule, time between doses, adjustments of weight, age, meals, treatment periods. Ideal clinical research protocol should explain about the dispensing method, administration instruction, supply of drugs, storage, study drug accountability, measures of treatment compliance. Thank you.